Now I got all the buttons clicked. Okay. So we're going to take a look at objective 3. We're actually not going to do objective 4 today. We're just going to do objective 3. Because for some reason my night class ended up taking up an hour and a half and we only did objective 3. So it actually didn't take that long, but we kind of got off topic. A few terms that you're going to need to know is muscle fiber. Again, that's also a muscle cell, but sarcolemma. Sarcolemma is a fancy name for plasma membrane of a muscle cell. So it's represented by this blue right here in this muscle cell. Again, it's just the plasma membrane, phospholipid bilayer, the whole bit, but it's a little special, so that it's got its own name. Anytime you see the prefix sarco, that means muscle. Okay, so that's referring to something going on with the muscle cell. T-tubules. Now, this one you're definitely going to need to know the definition of. So with T-tubules, if you think about it, this is your muscle cell, and you've got your action potential coming from your motor neuron that is going down the plasma membrane of that muscle cell. And that's all well and good, but what's actually doing the contracting of this muscle cell? It's the sarcomeres located within the specialized organelle called the myofibril. That's what's doing the contracting. So the action potential has to reach the myofibrils. Well, if the action potential is just running along the outside of the cell, it's not going to be able to get to the myofibrils on the inside. So if you notice, the plasma membrane, or sarcolemma, as we just learned it was called, has got these little holes in it. Those little holes are T-tubules, and that action potential, as it travels down the plasma membrane, will be able to go deep inside the cell, allowing the action potential to reach those myofibrils on the innermost area of that skeletal muscle cell. So when the action potential arrives, it's not just the myofibrils that are in contact with the plasma membrane that are going to get to contract. It's every myofibril within that skeletal muscle cell that's going to get to contract at the same time, because of the T-tubules, which is a passageway, allowing that action potential to get to the deep inside part of that skeletal muscle cell. That's how that gets in there. Yep, otherwise it just travels around the surface. Sarcoplasm, fancy name for cytoplasm, but because it's sarcoplasm, you know it's the cytoplasm of a muscle cell. When are we really going to talk about sarcoplasm? Probably not ever, but if you see it like embedded in a test question somewhere, you know, okay, that's just cytoplasm. I just need to go on like I know it's cytoplasm. So that's all that is. It's just so if that word pops up, you recognize it. Okay, that was actually where we were supposed to end day 1, but that's okay. Now, myofibrils, today we're really going to focus on the structure of the myofibril, the organelle that's found inside of skeletal muscle cells, specifically the actin and myosin that make up that organelle. So actin and myosin are both proteins. Actin is a thin protein. Myosin is a thick protein. And you can see right here, you've got your sarcolemma, which is the plasma membrane there. You can kind of see it kind of flapped over there. You've got your myofibrils, which run the length of the skeletal muscle cell. They look like a bunch of pipes that are just crammed into that skeletal muscle cell, and they all are contracting. So the more you use your muscles, the more myofibrils that muscle cell will have, meaning it's going to be able to work better. But we're going to focus on the specific parts that make up that myofibril, the actin and myosin. So here we're going to kind of stop the PowerPoint, and if I can get that button to click there, there we go. And we're going to do a little drawing. We're going to use about 4 pieces of paper. I'm going to use about 4 pieces of paper today. You may want to use more than that. I'm just going to use 4. So we're first going to take a look at the actin, then we're going to take a look at the myosin. So this one is the actin. Remember, it's the thin myofilament. And again, it's just a protein. made by the ribosomes at the direction of the DNA inside your nucleus. I mean, so your body makes these all the time, okay? Now, actin is made up of 3 smaller proteins that make this big, huge, thin actin molecule up. And we're going to take a look at each one of these actin proteins individually. So the first one we're going to look at is called F-actin. And it kind of stands for filamentous actin. So that's kind of where the F comes from. So I'm going to kind of draw that out, and it's supposed to be double-stranded. But I don't draw double-stranded well, so you're just going to get the single strand here. So there's our F-actin molecule, and F-actin has got what's called an active site. Now, this is a protein. You know, F-actin is a smaller protein that's making up the larger actin protein. But proteins have active sites. I've never mentioned them before because we've never needed to talk about it. But the job of the active site is where myosin binds. So it's the binding site for myosin. Actin and myosin are going to want to stick together, and this is the location where that happens. Just kind of on a side note, in case you see it in writing, which we're probably going to see here in a little bit, just to not be confusing, there is a thing called G-actin. Again, it's still got its little active site. The only difference is the G stands for globular actin. And this is basically just a single. See where the filamentous actin is many of these globs stuck together? The G-actin is just an individual. As far as I'm concerned, I am using these terms interchangeably. So if you see G-actin versus F-actin, just say, okay, it's the same thing. I'm not differentiating. I'm not going to ask you about it. But if you see it in writing, you're like, well, what's the G doing there? What's that? It's just really referring to, okay, it's an individual one is all it is. But I don't want you to get confused if you see a picture and it says G-actin versus F-actin. It's interchangeable as far as I'm concerned. Okay, so F-actin is where the myosin actually binds. Now, let's take a look at the second protein that makes up the actin molecule, which is going to be tropomyosin. Now, the job of tropomyosin is to cover or expose the active site on F-actin. So it's either going to cover it up or it's going to let it be visible. So we're going to look at 2 scenarios. We're going to look at our first scenario where it is covered, and then the second scenario where it's exposed. So in order to do this, we're going to draw 2 pictures. So we're going to take a look at our F-actin molecule with our active site. Y'all are going to get really comfortable drawing F-actin because we're going to draw it in all 3 pictures here. Now, in this one, we're going to cover the active site because we said it was covered. So there's our tropomyosin molecule. Just covers it, covers the active sites. So what does that mean? When tropomyosin covers the active site on F-actin, it prevents myosin from binding, right? Because if myosin wants to bind to the active site, but you got the active site covered up, myosin can't bind to it. So what is the end result? What does this mean? If myosin can't bind, the muscle will not contract, meaning the muscle is actually relaxed. So if your active sites are covered, you do not get a contraction out of that.
So they're not always.
They're not always covered because remember they can cover or expose the active site. So guess what B is going to be? Our next picture here, we're going to have an exposed active site. So we're going to draw our F-actin again. With their active sites. But this time, our tropomyosin is not going to be covering the active site. So what does this mean? Okay. When tropomyosin exposes the active site on F-actin, it allows, say if it's covered, it prevents. If it exposes, it allows myosin to bind, resulting in a muscle contraction.
What makes it covered and not covered?
Good question. We're coming to that next. I know you're getting there. You're thinking ahead. I know there's got to be a reason why we're covering and then covering this thing. So what's causing that? So bottom line is, if it's exposed, turning that up, y'all look like you're cold. If it's exposed, we're going to get a muscle contraction out of it. If it's covered, if the active sites are covered, we're not. Okay, so now let's take a look at, that was 2 of the 3 proteins that make up actin. So let's take a look at the third that makes up actin. And this one is called troponin. And I'm just going to call it the troponin complex because it's made up of 3 separate proteins. I know.
So it's
I know.
3 that's made up of
3. I know. It's not. I know. It'd be easier if it was made up of something different, but it's made up of 3 smaller proteins. And troponin acts like the glue that holds the actin together. So our three molecules here are troponin T, troponin I, and troponin C. So you need to know what each one of these binds to. Troponin T binds to tropomyosin. Remember, troponin is the glue, so it's holding everything together. So the molecule T connects to the tropomyosin molecule. The troponin I binds to the F-actin molecule. And the troponin C, you're like, well, we only talked about two things. What the heck is the C binding to? It binds to calcium. Wow. Kind of came out of nowhere. If you all remember when we talked about the skeletal system, we mentioned the importance of calcium, not only for the skeletal system, but its involvement in many organ systems. And that's why you have to have a certain amount in your blood at all times. So all these organ systems can have access to this calcium. And that's why calcium homeostasis is important. One of those organ systems that is required to have calcium in order to function is your muscle system. So you've got to have calcium in order to get your muscles to work properly. So let's take a look at how this troponin is all set up. So we're going to draw our F-actin molecule. We're going to draw our active site. Now, I'm not going to draw the tropomyosin just yet, which is the strand that either covers or exposes the active site. So I'm going to pause on that, and we're not going to do that just yet. But I am going to go ahead and draw the troponin complex, which is three molecules. Some people in the other class said it kind of looks like a snowman the way I did it. So, okay. So you've got your three molecules that represent your troponin complex. I'm not going to worry about which one is which. We're just going to say those three structures, each one represents the T, the I, or the C molecule. Okay, so now we have this surprise molecule that comes in called calcium. So we're going to add some calcium into our picture here. And remember, part of that troponin molecule binds to calcium. So if calcium is present, it's going to stick to our troponin complex. So it's a little snowman with a hat, I guess, if you want to think about it that way. And just to put this all back into perspective, this is part of the actin molecule, which is making up the organelle, the myofibril. So this calcium is inside of that myofibril organelle. It's present in there, and that's why it's able to bind to the troponin. So the calcium ended up inside the myofibril organelle, so it could bind to the troponin complex. So when calcium is present, it binds to troponin C, which causes the troponin complex to change shape. So it's one shape when the calcium is not bound. As soon as the calcium shows up and binds to the troponin C, the molecule changes shape. Now keep in mind, this troponin complex is attaching to everything. So if it changes shape, everything else has to shift. It has to, right? So if the troponin complex changes shape, this results in the tropomyosin moving, which will expose the active site on F-actin. So now I'm going to draw my tropomyosin in, and we know that the tropomyosin is not covering the active site because calcium is present. So you ask for what causes the tropomyosin to move? The presence of calcium.
So no calcium, it's covering calcium, it is not.
Correct. So when you've got your active site on the F-actin exposed, the myosin will bind to the active site on F-actin, resulting in a muscle contraction. So bottom line, calcium is present. You're going to get a muscle contraction out of that. Calcium is the trigger that causes that muscle contraction. No calcium, no muscle contraction. Just so I don't have to keep saying myosin binding to active site on F-actin all the time, I'm going to give you all a new word, a new phrase. It's called cross-bridge formation. We're going to talk more about this next time. And all that means is the myosin is binding to the F-actin molecule. So that's all we're saying. So instead of saying that big, huge, long phrase, that's mostly referred to as cross-bridge formation. Okay, wow. So those are your three molecules that make up the actin thin filament. Now let's take a look at the myosin filament. Does anybody have any questions about that before we go on to the myosin? Okay. This is the thick myofilament. And again, this is a protein that is created by at the instruction of the DNA inside the nucleus by the ribosomes, so all that good stuff. Okay, I'm going to go a little down the page here because we're going to put something up top, so I'm going to go a little bit further down. So we've got our myosin here. I'm going to draw this like super big. That's about it. So not too bad. This is the myosin tail, sometimes called the rod, part of the, kind of looks like a golf club that somebody's taken two big bites out of. And then we have our myosin head. Now this big bite that's been taken out of that myosin head, This is the active site. So this is where myosin binds to the active site on F-actin. So if I were to draw an F-actin molecule, it's a little bit bigger than what we're used to drawing, right? We were drawing smaller circles, but there's our F-actin molecule there, and here's our active site here. So this, because you have binding here, that's cross bridge. Right, because you have the F-actin bound to the myosin, so that's a cross bridge, and it's the active side of one meeting up with the active side of the other. Okay, so what is this other little bite that's been taken out of down here? This is the ATP, remember that word, ATP binding site. It contains ATPase, which is an enzyme that breaks down the ATP molecule. Remember, anything that ends in ACE is an enzyme. That, you know, it's an enzyme if it ends in ACE. So this is where ATP comes in and binds to the whole molecule. This area right here, where the head attaches to the rod, this is the hinge region. This has the ability to flex and extend. So that hinge will flex and it will extend. So it moves. That's what a hinge does. You've got movement there. Okay, so does anybody have any questions about, that's it, by the way, that's it for myosin. So why is it called the thick filament? That doesn't look very thick. What's going on? Why is it considered the thick filament? Because you've got about 100 or 300 of these all connected together. So in other words, you've got about 300 of these rods all connected, all kind of together, like if you had a bundle of straws or something like that. So it makes it a thick filament. Individually, it's thin, but when you put about 300 of them together, boom, now it's thick.
So the F-actins go towards that?
When the active site is exposed, actually the myosin goes towards the actin. The actin doesn't do a lot of moving except just exposing the active site. The myosin does the majority of the moving here, okay? And it goes towards the actin when that active site is exposed. Okay, so now, because I think at one point I failed to go over this ATP breakdown business here very well. I think I went over it, but it was super duper quick. And I want to say it was a day we were virtual because we didn't have electricity here or something was going on. We had a down day one day. So anyway, I'm going to go over this ATP breakdown because we need to know it for next time. We're just going to kind of explain what that ATP breakdown is, and we're going to make it do stuff the next time we come in. So we've got ATP breakdown, also known as hydrolysis of ATP. Now, if you look at the word, hydro means water, lysis means to split. So what we're really doing is we're splitting that ATP molecule with the breakdown of water. You don't have to know where water, I'm not going to get into the water part. We're just going to do an overview real quick. But that's really why it's called hydrolysis of ATP, because you actually use water to break the molecule down. So let's first take a look at ATP. And it stands for adenosine triphosphate. So that's where those letters come from, adenosine triphosphate, ATP. Now I'm not going to draw the chemical structure of ATP. We're going to kind of, shorthand a little bit here. So this is the adenosine part of the molecule. And it's got 3 phosphates attached. So we're going to have a phosphate, and a phosphate. And if you all remember from chapter 2 or 3 or something like that, each one of these lines represents a chemical bond. And that bond is storing energy. Y'all might remember that word from IPC or physics or something. It's called potential energy. It's stored energy. As opposed to kinetic energy, which is energy of something moving. This is not moving. So these bonds, all bonds have stored energy. So what's going to happen is that ATPase molecule is going to break that bond holding the 2nd and 3rd phosphate together, which is going to result in a chemical reaction here. And when that bond breaks, releasing that 3rd phosphate, we're going to get a molecule that only has 2 phosphates. And that is called ADP, adenosine diphosphate, which stands for ADP. Now, according to the law of conservation of mass, you can't have 3 phosphates on one side and only 2 on the other. That's not going with the conservation of mass. You can't just proof magically the phosphate just went away. We don't know where it went. It's still there. I'm going to put PI over here, though, because it's considered an inorganic phosphate, and the fact that it came from the result of a chemical reaction. It wasn't like food that you ate or something like that. So, the other thing that we're going to make over here, this is an exothermic reaction, which means when that bond broke, it released a ton of energy. So that bond breaking, that potential energy went into kinetic energy, basically. We converted our stored energy into energy that's going to do something. Now, for example, in our myosin head situation, that energy will be absorbed by the myosin head, because energy can't be created or destroyed either. We can't just disappear. It has to go somewhere. So it's going to absorb into the myosin head, causing movement. So it's going to cause that myosin head to move. That's what that energy is going to do when it gets absorbed into the head. Now, it's going to do something different with the sodium-potassium pump, right? We need an ATP for that as well. So when the ATP breaks down and gets absorbed into the ATP pump, or the sodium-potassium pump, it causes the pump to change shape. So again, it causes a shape change. That energy results in a shape change of the protein when that ATP molecule breaks down. So we're not getting into the details. I mean, that's pretty rough overview there. We do need to understand a little bit about the hydrolysis of ATP, the breakdown of ATP, when we get into next time. So this is just, again, all we're really going to worry about. We're not going to get into where water comes in or any of that kind of stuff. Okay, so does anybody have any questions about that you think of?
When the ATP comes in, breaks off that last one, moves it into ADP.
Now you got some ADP floating around inside the cell. And it doesn't contain as much energy. Your ATP molecule is a high energy molecule.
So,
ADP has a little bit of energy, but it's just technically not used a lot. So we've got our, just to kind of go over the PowerPoint, it's all in here, so I'm not telling you anything that isn't already, that we haven't talked about. So you've got your actin, which is composed of your F-actin molecule, your tropomyosin molecule, and your troponin molecule. You can see over here, see it's called G-actin here. It just means the individual ones, but the whole strand is F-actin. And then your tropomyosin is the rope-like structure that's covering the active site. And then you've got your 3-molecule troponin complex there as well. Okay, so the F-actin, just kind of talking about that individually, it's the one that has the active site. You can kind of see that active site in that little yellow circle right there that's sitting on top of the red ones. And again, you can see the G-actin, which is just referring to an individual over here. But this is the active site where the myosin binds. Now with the tropomyosin, you can see it represented by a blue strand, and you can see it definitely covering the active site. And that's why myosin is not able to attach, because the tropomyosin is in the way. But here comes the calcium, that little black one right there, that's the calcium. Here comes the calcium. It's fixing to bind to that troponin complex. So the troponin, remember, is made up of 3 parts. You've got the troponin I, which binds to the F-actin. That should be F-actin right there. You've got troponin T, which is going to bind to the tropomyosin molecule. And you've got your troponin C, which is going to bind to the calcium. So here, it's kind of hard to see in this one where that tropomyosin complex is, but it's right here. There's that tropomyosin complex right there. You've got your T, your I, and your C, tick, if you want to try to remember what those letters are. You can see that the calcium is not bound, so we don't have cross bridge formation. But you can see in this one, it is bound, so we do have cross bridge formation. So the myosin is now able to bind to that actin. So with the myosin, again, you've got your tail and you've got your head. And this right here is kind of what I was trying to explain. It's about 300 individual myosin molecules all stuck together. Notice the heads are pointing outward. None of the heads are going in. Why would that be? The head has to attach to the actin. It'd be kind of silly to try to attach it if you're hiding it in the center of the molecule. That's not going to work. So you got to have the heads pointing outward with all the tails going in. And it's a parallel structure, right? It's all running horizontally. This right here is, I would say, the big thing you need to know about your myosin heads. I don't know what slide number that is, but we've already talked about it, but if we're putting it all on one slide here, the 3 important properties of your myosin heads. First, the myosin heads bind to the active site on the F-actin molecule to form those cross bridges. Second, the myosin heads are attached to the rod portion by a hinge that is allowed to flex and extend, bend and straighten during a muscle contraction. And then third, the heads contain that ATPase enzyme, which breaks down, releasing energy, causing that to bend in the hinge region. So that's where that movement comes from. So these are 3 important properties of those myosin heads that you need to know. And we talked about them individually. We just didn't put them all on the same slide. Now I want to show you this animation so you can visualize, again, visualizing how this all works is difficult. I get that. So we are, keep in mind, we're within inside the organelle, the myofibril. We're inside that specialized organelle, and that's what's making up this actin and myosin. That's where it's found. So this is your myosin. That pink is the myosin. This green up here is the F-actin. So yeah, we're not seeing all of the rest of the actin molecule, the tropomyosin and the troponin, because all this animation is showing you is how the myosin attaches to the actin, and it only attaches to the F-actin, so that's why you're seeing that. So why are some of the F-actin molecules blue while the rest of them are green? The blue ones are where the cross bridge is going to occur, so that's why they're green. You're not seeing the active sites or anything like that.
Myofilaments are composed of strands of the proteins actin and myosin. An actin filament has two strands of actin molecules strapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head fluxes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This brings the myosin head to bind with a different actin molecule part of the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Okay, so did y'all catch what the ATP's job was? The myosin pulls the actin, but unless it's got ATP, you don't get cross-bridge release.
Won't let it go.
So you can get the muscle contraction, but ATP is what causes the muscle to basically muscle relaxation. And have y'all ever heard of rigor mortis? What happens during rigor mortis? It happens when someone dies. So what happens to their body during that phase? They stiffen up, right? They completely get stiff. So it takes 12 hours in a normal environment for them to completely, with all their muscles, to completely contract and stay contracted. And that stays that way for about 12 hours. And then all of a sudden, the muscles relax. So what's going on? What do you not have when you're dead? Well, you don't have oxygen, you don't have energy. You can't make that ATP molecule. So what happens is the muscles can contract, but they can't relax because there's no ATP around. So that's what happens during rigor mortis. So why is it that you're in complete rigor for about 24, during the 12 to 24 hour mark after death? During the first 12 hours, you get into rigor. And then from 12 to 24, you're completely stiff. And then from 24 to 36, you start to relax again. So where'd the ATP come from? I mean, obviously you had to have some, right? Or they wouldn't have been able to relax. Well, actually, not to be kind of morbid, but the bacteria are actually breaking down the muscle fibers. So that's what's breaking the cross bridges. It's the bacteria acting on the body and decaying, it's body decay. So it's not that all of a sudden you showed up with ATP. It's bacteria consuming the body. And this is how they can determine time of death. So if you ever wondered how they get time of death, that's kind of how they do it. They look to see what stage of decay the body is in, and they can back it up because they know under certain environments, the body's going to behave a certain way during decomposition. Have y'all ever heard of body farms? There's body farms. There's one in Tennessee, and there's one in San Marcos at is it Texas State University that's down there? And what they do is people who donate their body to science, some of them go to these body farms. And what the people do that work there, they put these bodies, these dead bodies, in different environmental situations to determine the time of decay for each, because it's a process. So how long does each process take if the body is in this environment versus this one? Are we in a high moisture environment, low moisture environment? Are we underwater? Are we, buried in the ground? Are we sitting on top of the ground? Is it cold? Is it hot? Is it humid? So they have bodies in all different kinds of environments, and that's how they can tell time of death, and they can back that up and be fairly accurate because, again, in a different environment, the body's going to act a different way. Somebody was telling me in the last class, in the 6 o'clock class, they said, yeah, I was reading a story about a killer who killed somebody and put the body in a house and turned the air conditioner way up. So it was freezing in the house. Well, why did they do that? Well, to delay the decay of the body so it doesn't stink and all that kind of stuff. So obviously their time of death would be different. I mean, you would have to calculate it differently than a person who is in a hot, humid environment. That decay process is going to happen more rapidly. So when the medical examiner comes in, they can kind of guesstimate time of death based on the environment that the body is in and the rate of decay at that time. And then you include all the bugs that show up as well. I mean, that happens too. What bugs are present and at what stage of their life cycle are they in? So it helps determine time of death. So we kind of got off on a tangent there. Sorry about that. But what we're going to look at now, again, we're almost done. What we're going to look at now are the sarcomeres. Now, keep in mind, we are looking at the myofibril, which is within the muscle cell. You can see there's a lot of myofibrils in there, and that myofibril has got sarcomeres that go end to end to end all the way down that organelle. So remember, the sarcomere goes from one Z disc or Z line to the other Z line. So that's the sarcomere right there. And this muscle is relaxed. Now, in the contracted muscle, you can see you've got your Z line or your Z disc to your other Z line or your other Z disc, and you can see that sarcomere is a lot shorter. Remember what we said the other day. The sarcomere is the smallest contractile unit of a muscle cell. So when your muscle is contracting, it's actually the sarcomeres that are getting smaller. So which causes the whole muscle to get smaller or contract. So let's take a look at what's going on here, so we can kind of see a little bit more of what's happening. We've got our actin. You'll see these little purple lines. Those are all your actin thin filaments. And then you can see these red ones, which are going to be your myosin. Okay, and remember I said the actin and the myosin are actually parallel to each other. Okay, so in a relaxed muscle, what you're seeing, let me get my highlighter on here going, you can see there's not much overlap between the actin and the myosin in a relaxed muscle. The actin and the myosin are spread really far apart. But in a contracted muscle, look at the overlap of the actin and myosin now. They have a lot of overlap. So if you want to think about it, in a relaxed muscle, the actin and the myosin are spread really far apart. But in a contracted muscle, that myosin pulls the actin, so they overlap more. So it shortens the sarcomere. So that's kind of what's going on visually. I get that's kind of hard to see. You can see the individual little heads here of those myosin molecules sticking up, ready to grab that actin molecule. And we will come back next time, take a look at objective 4, and kind of put all this into action so you can see the myosin grabbing the actin and pulling it along, causing the sarcomere to shorten. And we'll also take more look at the answer to the essay question Tuesday as well. So we just kind of laid out all the parts today with all of their jobs, and then kind of like what we do with the nervous system. And then next time we're going to put everything into action and get it all working.